Veterinary physiology, brain teasers. Silent points. Cattle are more prone to retrograde drainage from pharynx because they have relatively small lungs with low tidal volume and residual capacity. Tonsillitis is common in dogs but rare in cats. Tonsillitis is the disease of dogs. Rectal temperature in increasing order. Maximum rectal temperature is of horse and minimum rectal temperature is of a rabbit. And rabbit, horse and cattle have maximal rectal temperature. Pig and rabbits have the minimal temperature, and the order is as maximum horse, then cattle, then dog, sheep, pig and rabbit. Heart rate in decreasing order. The maximum heart rate is of mouse and chicken, maximal of mouse then chicken, and the minimal heart rate is of ruminants as sheep, goat and cow. Minimum heart rate is of sheep, goat and cow. Maximal is of mouse. And their heart rate sequences as maximal mouse, chick, guinea pig, rabbit, cat, pig or dog, sheep, goat, dairy cow, ruminants. And increasing order of urine volume is as follows. Maximum urine volume ml per kg is of dog. Dog has a maximal urine volume as compared to its body weight, and horse and pig has the minimal urine volume as compared to their body weight. Dog and cow have the maximal urine volume, and the minimal urine volume is of horse and pig. The sequence is as horse, pig, cat, small ruminants, and then dog. Horse, pig, cat, ruminants, and then dog. And the increasing heart rate is as mouse, chicken, guinea pig, rabbit, cat, pig or dog, and then ruminants. And rectal temperature in increasing order is as maximal is of rabbit, then ruminants, and then horse. Urine of cow is more viscous than cat. Cow urine is more viscous. Alanine aminotransferase is a liver specific hepatocellular enzyme in dogs, cats, and primates. Primates as humans. Dogs, cats, and primates have an alanine aminotransferase liver specific hepatocellular enzyme. Alanine aminotransferase is in dog, cats, and primates. Serum amylase activity increases within 12 to 48 hours after pancreatitis. In pancreatitis, serum amylase activity increases within 12 to 48 hours. So the pancreatitis can be detected with increasing serum amylase after half day of infection. Stratum corneum is compact layer of dead cells in order to prevent loss of fluids, electrolytes and water. In order to prevent loss of fluids, electrolytes and water, stratum corneum is a compact layer of dead layers. Stratum corneum is a dead cell layer. Its function is prevention of loss of fluids, electrolytes, and water. Langerhans cells are mononuclear dendritic cells that are intimately involved in regulating immune system of the skin. Langerhans cells are mononuclear dendritic cells that are intimately involved in regulating immune system of skin. The immune system of skin is mediated by or controlled by Langerhans cells. Except in horses, apocrine glands do not appear to be innervated. Apocrine glands are not innervated. They are only innervated in horses. Hair follicles of dog, cat, sheep, and goats are compound. Sheep and goats. Small ruminants, canine felines have compound hair follicles, where cattle and horse, the large animals, have simple hair follicles. Small ruminant cat dog have compound hair follicles, whereas the hair follicles of cattle and horse are simple. Growing stage of hair is known as anagen. Anagen is the growing stage of hair. Resting stage of hair is telogen. Telogen is the resting stage. Anagen is the growing stage. Anagen growing stage. Telogen resting stage. Thyroxine initiates and glucocorticoids inhibits the hair growth. Hair growth is initiated by thyroxine. So thyroxine can cause hyperkeratosis or anagen, and glucocorticoids inhibits the hair growth, that is also known as the telogen. And hairs of cattle and horse are have simple follicles, whereas that of small ruminants and dogs have compound follicles. Paniculus adipose serves many functions like insulation, reservoir for fluids, and shock absorber. Paniculus adiposus. Paniculus adiposus serves many functions like insulation, reservoir from fluid for fluids, and shock absorber. Abomasum is not a part of forestomach, and parts of forestomach are rumen, reticulum, and omasum. Abomasum is true stomach or glandular stomach. Gizzard is present in poultry and it is known as muscular stomach. Proventriculus is glandular stomach. Gizzard is muscular stomach. Proventriculus is glandular stomach, and both are present in poultry. Bursa of Fabricius is immunological organ for poultry. Poultry has immunological organ as bursa of Fabricius. The term physiology was introduced by Jean Fernet. Fernet used the term physiology by Jean Fernet. The functional unit of body is cell. Basement of is composed of mucopolysaccharides. Basement is composed of mucopolysaccharides. Karyoplasm is contained in nucleus. Nucleus has karyoplasm. Nucleus was discovered by Brown, and cell was discovered by Robert Hooke. Fernet Jean Fernet used the term physiology, coined the term physiology, and Robert Brown discovered the nucleus, and nucleus has karyoplasm. Mucopolysaccharides makes the basement, and functional unit of body is cell. Process of synthesis of yolk is called vitellogenesis. Vitellogenesis is the process of synthesis of yolk. Messenger RNA is synthesized in nucleus. Messenger RNA is synthesized in nucleus. Respiratory pigment of blood is hemoglobin. Hemoglobin is the respiratory pigment of blood, and messenger RNA is synthesized in nucleus. Vitellogenesis is the process of synthesis of yolk, and nucleus was discovered by Robert Brown. Karyoplasm is contained in nucleus. Basement membrane is composed of mucopolysaccharides. Functional unit of body is cell. Physiology term was introduced by Jean Fernet. Nucleus was discovered by Robert Brown. Robert Hooke discovered the cell, and Jean Fernet coined the term physiology. Vitellogenesis is the process of synthesis of yolk, and the process by which a more diffusible materials can separate from non-diffusible materials is known as dialysis. Dialysis is the process by which more diffusible materials can be separated from non-diffusible materials. True oxide of hemoglobin is methemoglobin. Methemoglobin is the true oxide of hemoglobin. Dialysis is the process by which diffusible materials can be separated from non-diffusible materials. Monocytes are produced from myeloid tissue or bone marrow. Platelets are produced from myeloid tissue or bone marrow. They give rise to platelets and monocytes. True hemoglobin is methemoglobin. Monocytes and platelets are made from myeloid tissue. The structural and functional unit of compact bone is osteocytes. Osteocytes are the structural and functional unit of compact bone. Fructose is absent in seminal plasma of dog. Secretion of seminal vesicle is acidic and the seminal plasma do not have fructose in case of dogs. Luteinizing hormone surge is responsible for ovulation within 6 hours. After 6 hours of LH surge, there is ovulation. Normal hemoglobin concentration in chicken blood ranges 6.5 to 9 gram per 100 ml. In which animal species corpus luteum is not maintained in pregnancy? In mare, corpus luteum is not maintained in the ovary, whereas the source of progesterone is also the placenta. So after if there is no corpus luteum in ovary, then there is still the pregnancy not uh, proceeds as abortion, as there is another source of progesterone too, that is placenta. Hemoglobin concentration in chicken is 6.5 to 9 gram per 100 ml. Luteinizing hormone is responsible for ovulation within 6 hours and secretion of seminal vesicles is acidic. Fructose is absent in seminal vesicle of dog. Structural and functional unit of compact bone is osteocytes. Platelets are produced from myeloid tissue. Monocytes are produced from myeloid and bone marrow. Myeloid or tissue or bone marrow, they give rise to platelets, and monocytes. Two oxides of hemoglobin are methemoglobin. Dialysis is the process by which more diffusible materials can be separated from non-diffusible materials. Prostaglandin is the fetal hormone that is responsible for initiating parturition in dam. The hormone preferred for estrus synchronization in doe is prostaglandin. Prostaglandins are the hormone preferred for estrus synchronization in doe in sheep. It is available in Pakistan as cyclomate. Prostaglandin is available as cyclomate, and GnRH are available as conceptal. The fetal hormone prostaglandin are responsible for initiating of parturition, and they are used in estrus synchronization. Pheromones secreted in saliva of which species attracts females and induces immobilization reflex. In boar, pheromones are secreted in saliva and it attracts females and induces immobilization reflex. Pheromones of the saliva of a boar attracts females and they induce immobilization reflex, and prostaglandin are used in estrus synchronization and initiation of parturition. Vagus nerve fibers are mixed. Vagus nerve is a mixed nerve. Their fibers are mixed nerve fibers. Which of the following events take place earlier in the process of parturition? Increase of prostaglandin level. Increase of prostaglandin level is the one of the event that takes place earlier in the process of parturition. And vagus is mixed nerve. Boar saliva has pheromones that attracts female and induce immobilization. Prostaglandins are used for estrus synchronization and are responsible for initiation of parturition. pacemaker of heart is SA nerve node. Sinoatrial node is the pacemaker of heart. The third heart sound is produced by closure of atrioventricular valve. The closure of atrioventricular valve is responsible for the third sound of heart, and sinoatrial node and SA node is the pacemaker of heart. The size of red blood cells is smallest in sheep. Option here is not goat, otherwise goat may have the smallest size followed by the sheep. So the size of red blood cell is smallest in sheep and largest is in camel. Camel shape is also different. The third heart sound is by atrioventricular valve and sinoatrial node. Sinoatrial node is the pacemaker of heart. Increased prostaglandin levels are reported in earlier process of parturition and estrus synchronization. Saliva of boar has pheromones to attract females, and initiation of immobilization. Reflex vagus nerve is mixed type of nerve, and the size of red blood cell is largest in camel, smallest was in sheep. Smallest red blood cells are in sheep or goat and largest in camel. Also red blood cell also has the different shape, and nucleated red blood cells are in avians, and the mature red blood cells of mammals do not have nucleus, and the cat may have some of the precursors of red blood cells that may be nucleated, and that may be that may indicate the initiation of any infection etc. So it may be the physiological phenomena if the cat have the nucleated red blood cells, as they are the precursors of the mature red blood cells. Precursors of red blood cells are nucleated, but in mammals after at maturation or maturated red blood cells they are enucleated. Hypochromic red blood cells means hypo mean less, chrome mean color is by hemoglobin, so hypochromic mean less color, less hemoglobin, less concentration of hemoglobin per red blood cell. In methemoglobin, iron remain in the form of Fe+3. Fe+3 is the methemoglobin, and hypochromic red blood cell mean less concentration of hemoglobin. Which of the following anticoagulant acts by accelerating the activity of antithrombin 3? Heparin is an anticoagulant that acts by accelerating the activity of antithrombin 3. Heparin activates antithrombin 3. Heparin is an anticoagulant that accelerates antithrombin 3. Antithrombin 3 means no thrombus formation, no blood clotting. The responsible agent is heparin, so it is the anticoagulant. Heparin is also the anticoagulant present in the saliva of mosquito. In methemoglobin, iron remain in the form of iron in the Fe+3. Lesser concentration of hemoglobin per red blood cell is known as hypochromic. Smallest red blood cell is in sheep and largest is in camel. And sodium EDTA acts in association with calcium. Sodium EDTA, that is an anticoagulant, acts in association with calcium. Sodium EDTA binds calcium. The precursor of platelets is megakaryocytes. Megakaryocytes are the precursor of platelets, and the clotting factor of vitamin K is inhibited by decomerin. The clotting factor of vitamin K is initiated by decomerin. Decomerin inhibits the clotting factor vitamin K, or decomerin inhibits vitamin K. Vitamin K is inhibited by decomerin, and megakaryocytes are the precursor of platelets. Sodium EDTA binds calcium, so blood cannot clot, and heparin activates antithrombin 3. Two anticoagulants as EDTA and heparin. Heparin accelerates antithrombin 3 activity, whereas sodium EDTA binds calcium. And iron plus Fe plus 3 is methemoglobin, and in clotting factor vitamin K is inhibited by decoumarin. Decoumarin inhibits the vitamin K clotting factor, inhibits vitamin K. Hinge bodies are clumps of denatured hemoglobin. Hinge bodies are the clumps of denatured hemoglobin, and Howell Jolly bodies are the remnants of nucleus. The two anomalies of red blood cells, Howell Jolly body having the fragment or any nuclear remnant within the red blood cell, and if there is denatured hemoglobin in red blood cell, then it will be the Hinge body. Hinge body is the red blood cell having denatured hemoglobin, and Howell Jolly body is the red blood cell having remnants of nucleus. As the red blood cell in its primary stages or developing stages, it has the nucleus, but it becomes anucleated upon its full maturation. So during this process, if any of the nucleus remained or fragment of nucleus remained in the red blood cell, then it will be the Howell Jolly body. And if this fragment is by the denatured hemoglobin or the red blood cell having a denatured hemoglobin, then it will be the Hinge body. Schistocytes are red blood cell fragments. Schistocytes are also the red blood cell fragments. There are three. Till now we have three main anomalies of red blood cell, as Howell Jolly body, as the nucleus having the remnant, Hinge body having denatured hemoglobin, and schistocytes are the fragmented red blood cells or the red blood cell fragments. Till now we had studied that the fetal membrane is responsible for prostaglandins initiate parturition. They are used for stress synchronization, and the pheromones of bull the pheromones are in saliva of bull that attracts females and induces immobilization reflex. Vagus nerves are mixed type of nerves, and pacemaker of heart is called to the sinoatrial node. Third heart sound is produced by atrioventricular valve. Smallest size of red blood cell is in sheep, and the cat may have physiologically normal red blood cells with remnants of nucleus, and camel has the largest sized red blood cell. Shape of camel is also different from other cells. I think the shape of camel red blood cell is biconvex, but not sure. Hypochromic red blood cell mean red blood cell with less chrome or less hemoglobin. Methemoglobin mean iron plus 3. Which of the following anticoagulant? Heparin accelerates. Heparin and EDTA, sodium EDTA, both are anticoagulants, and their mechanism of action is as follows: as the heparin binds antithrombin 3, and the sodium EDTA binds calcium. Both results in the anticoagulation of the blood do not clot. Heparin is the natural anticoagulant present in the saliva of mosquito, whereas sodium EDTA is commonly used in labs as anticoagulant. The precursor of platelets is megakaryocytes differentiates into platelets. Decoumarin is the inhibited by vitamin K. Vitamin K, or the antagonist of vitamin K, is decoumarin. Decoumarin decreases vitamin K. So if anyone have the deficiency of, if anyone have very much vitamin K, then he may be subjected with decoumarin. Hinge bodies are clumps of denatured hemoglobin. If the red blood cell has denatured hemoglobin, then it will be the Hinge body, and if the red blood cell have the remnant of nucleus, then it will be the Howell Jolly body, and if red blood cell is fragmented, then it will be the schistocyte. The size of caprine erythrocyte is 16 micrometer. Oval jelly bodies are found in red blood cell. Red blood cell having the nuclear remnant are called as the oval jelly bodies. And the other condition of red blood cell we had studied till now are the Hinz body, as they are the denatured hemoglobin. If the red blood cell have denatured hemoglobin, then it will be the Hinz body, and if the red blood cell has the nuclear remnant, it will be the oval jelly body. Red blood cell having degenerated hemoglobin, Hinz body. Red blood cell, nuclear remnant, oval jelly body. And schistocytes, red blood cells fragmented, are the schistocytes. Oval jelly bodies are found in red blood cells, and they are the red blood cell having the nuclear remnant. Erythrocytes are non-motile, they have no flagella, no cilia, they move on the direction of the flow of the blood. Which of the following is the percentage of erythrocytes in whole blood? Hematocrit is the percentage of erythrocytes in whole blood. The blood having the concentration of erythrocytes is known as the hematocrit or packed cell volume. Hematocrit is another name of packed cell volume, and it is the percentage of erythrocytes in whole blood. Which of the following is the associated with formation of erythrocytes? Red bone marrow is associated with the formation of red blood cells, as the red blood cells are formed in bone marrow, and the functional bone marrow or the active bone marrow or the bone marrow that is making the red blood cells, its color is dark red. So the reddish dark bone marrow is associated with the formation of erythrocytes. Variation in size of erythrocyte is called poikilocytosis. Poikilo means variable. Poikilocytosis is variable, or variable cytosis means cells. Variable size of cell is known as the poikilocytosis or picocytosis. Picocytosis can also be remembered with a fact that the mammals and the birds are homeotherms, and the amphibians are poikilotherms. Amphibians are poikilotherms, as poikilo means many, as the amphibians has many temperatures, and their temperature is as the temperature is of the external environment. If the external environment is cold, they decrease their or lower down their body temperature, and at hot conditions their body temperature is increased. So their temperature fluctuates, their internal temperature fluctuates, and it is not constant. So they are called as poikilotherms or picolotherms. Same is the case in the red blood cells. If the size of red blood cell is variable or non-constant, then it will be the picocytosis or poikilocytosis. The variation in size of erythrocytes is called as picocytosis. And anisocytosis is the deviation from normal shape of erythrocytes. If the red blood cell shape is different from that of normal, then it will be anisocytosis. Do not confuse shape with size. Size is a larger or smaller. If the cells are size is increased or decreased, smaller or larger, then it will be picocytosis. And deviation from normal shape of erythrocyte is anisocytosis. Red blood cell larger than normal is called as macrocytes. Macrocytes are the anisocytes that have the larger red blood cells. Macrocyte can be the type of anisocytes, as macrocytes means increased in in size of red blood cell, and anisocytes mean change in shape of red blood cell. Which disease is characterized by an increased bone turnover rate or accelerated bone formation and resorption? Pigot's disease. Pigot's disease involves the increased bone turnover, or the Pigot disease involves accelerated bone formation and reabsorption. Bone is formed and resorbed in Pigot's disease. Macrocyte is the largest sized red blood cells. Decrease in shape is anisocytes. Decrease in size is picocyte. Red blood cell is the red bone marrow is involved with the formation of red blood cells. Hematocrit is the percentage of erythrocytes in whole blood. Red blood cells are non-motile, and the size of smallest red blood cells are in sheep, and the size of caprine erythrocytes is 16 micrometer. Largest red blood cell are in the camels. Heme is porphyrin consisting of 4 pyrrole groups, and each pyrrole group binds in an iron molecule. So heme can bind 4 iron 4 iron Fe+2. Erythrocytes of mammals lose nucleus during maturation. At bone, within the bone marrow, or the precursors of the red blood cells are nucleated, but at maturation they do not have nucleus. But if any red blood cell have the nuclear remnant, then it will be called as whole jolly body. But some cat may have the red blood cells that have the red blood cells that have the nuclei, and it is a physiological condition in cat, as in some conditions erythrocyte cat as in some conditions cat have the very much increased rate of red blood cell formation, in which they have the premature red blood cells, and these premature red blood cells are nucleated. It is normal physiological condition. Erythrocytes of cattle, sheep, goat and pig are discoid. Disc shape is the shape of erythrocytes of cattle, sheep, goat and pig, and it is not the shape of camel. Camel are, I think, biconvex. Discoid is the shape of erythrocytes of ruminants. In a summary, canine erythrocytes is biconcave. Canine erythrocyte is biconcave, same as that of humans, but the shape of red blood cell of ruminants is different from that of the canines and humans, as the ruminant red blood cell shape is discoidal and that of canine is biconcave. In lymph nodes, T lymphocytes are largely found in parafollicular zone. Parafollicular zone is the zone in lymph nodes where T lymphocytes are formed. T lymphocytes are formed in the parafollicular zone of lymph nodes. Which of the following serves as carrier of hemoglobin? Hepatoglobin. Hepatoglobin serves as the carrier of hemoglobin. Which of the following serves as carrier of hemoglobin? Hepatoglobin serves as the carrier of hepatoglobin. Parafollicular zone is the zone of lymph node in which T cells are formed, and mammalian erythrocytes have every lifespan of 120 days, or 3 2 6 3 3 9 3 4 12. 4 months is the age of mammalian erythrocytes, and do not confuse with the same lifespan of erythrocyte with that of the shape, as they have different shapes. Biconcave shape is the shape of humans and canines, especially dogs, whereas that of the shape of ruminants erythrocyte is discoidal shape. Excluding camel may have the biconvex shape red blood cells. Closure of pulmonary and aortic valves are associated with second heart sound. We had studied the third heart sound, remember that, but the second heart sound is the closure of pulmonary and aortic heart valves. And granulocytes are formed in bone marrow. Bone marrow is associated with the formation of granulocytes, and red bone marrow is associated with the formation of red blood cell, or the red blood cell. More the reddish is the color of bone marrow, more active is the formation of red blood cells in that bone marrow. And shorter bones or small bones have more the percentage of formation of red blood cells. Longer bones or large bones have less proportion of formation of red blood cells. Closure of pulmonary and aortic valves are associated with second heart sound. Till now we have studied that bone marrow is associated with the formation of granulocytes. Second heart sound is the closure of pulmonary and aortic valves. 120 days is the lifespan of red blood cells. Hepatoglobin serves as carrier of hemoglobin. Hepatoglobin serves as the carrier of hemoglobin. And in lymph nodes, T lymphocytes are largely found in parafollicular zone. Parafollicular zone is the zone where T lymphocytes are formed and parafollicular zone is present in lymph nodes. Canine erythrocytes are biconcave and that of the ruminants are discoid. Erythrocytes of mammals lose nucleus during maturation. Heme is porphyrin consisting of four pyrrole rings, and each pyrrole ring binds an iron, so heme can bind four each iron combined with an oxygen, so heme can bind four oxygen molecules. Which disease is which disease has accelerated bone formation and resorption? Pigot's disease has accelerated bone formation and resorption. Macrocyte is the red blood cell larger than the normal shape is the macrocyte. Anisocyte is the change in shape of red blood cell. Picocytosis is change in shape of size of erythrocyte. Change in shape anisocyte. Change in size picocyte. Red blood cells shape is changed anisocyte. Size is changed picocyte. Red bone marrow is the area where red blood cells are formed. Hematocrit is the percentage of erythrocytes in whole blood. Red blood cells are non-motile red blood cells. The heminglia remnant is the whole jolly body. The size of caprine erythrocyte is 16 micrometer. Blood platelets are increased in anemia. In anemia, blood platelets are increased. A low platelets count is seen in thrombocytopenic purpura. Thrombocytopenic purpura leads to decreased platelet count, and increased platelet count is in anemia. Death in sweet clover disease is due to hemorrhage. Sweet clover disease leads to hemorrhages that ultimately leads to death, and anemia leads to increased platelet count. Thrombocytopenia purpura leads to low platelets count. The formula plasma volume into 10 by 100 minus 0.87 into hematocrit gives the blood volume. It's the blood volume, that plasma volume multiplied by 100 by 100 minus bracket 0.87 into hematocrit. Its resultant will be the blood volume. Hematocrit value will be approximately 30. 0.87 into 30 will be approximately 25. 100 minus 25 will be 100 minus 25 will be 75, and 100 divided by 75 will be around 2, and that will be multiplied with plasma volume to give the blood volume. At centrifugation of the blood, the plasma volume is approximately 50%. 50% of the total blood volume is plasma. In cattle, in normal conditions, in cattle, normal number of blood group systems found are 12. There are 12 blood group systems, and the formula of 100 by 100 minus 0.7 into hematocrit gives into plasma volume gives blood volume. Hemorrhage are observed in sweet clover disease that can cause death. Increased platelets are observed in anemia. Decreased platelets are observed in thrombocytopenic purpura. 12 blood groups of cattle had been found till to date. Which of the following helps heme to keep iron in ferrous form? Globin. The globin of hemoglobin keeps the iron in ferrous form. Which of the following is the organ of detoxification? Liver is the organ of detoxification. It is also the organ for first pass effect, that is associated with the per oral administered drug, food or toxic substances, and globin is associated with keeping the iron in ferrous form. Iron remains in ferrous form due to the globin of hemoglobin, that is the red blood cell pigment. There are 12 blood group systems in cattle, and it is the 4 times to that of humans. Humans have 4 blood group systems, AB and O, whereas cattle has 12, 4 times more complicated than that of humans. Which of the following is the organ of detoxification? Liver is the organ of detoxification. Eosinophilia is observed in parasitic infections, and bone marrow and brain are not supplied with lymphoid vessels. Bone marrow and brain do not have the lymphoid vessels. The lymph flows towards heart. The lymph flows towards heart. It means that it moves towards heart simply, which is not supplied with lymphoid vessels. Bone marrow and brain do not have lymphoid vessels. They are not supplied with blood vessels. Eosinophilia is observed. Increased eosinophil are observed in parasitic infections, and liver is the organ of detoxification. Iron remains in ferrous form due to globin of hemoglobin. 12 blood group systems are in cattle. Hemorrhage are resulted by sweet clover poisoning that may lead to death, and increased platelets are observed in anemia, and decreased platelets are observed in thrombocytopenic purpura. Smallest size of cecum is seen in dog. Maximum size of cecum is seen in horse. Horse has well developed cecum, whereas the regressed or smallest sized cecum is observed in canines or dog. Which of the following species has the minimum relative capacity of stomach in relation to body weight? Horse. Horse is the animal having 300 plus body weight, and it's usually for 500 kilogram, but it do not have the compound stomach. So simple stomach, only one compartment, very large size, that fits this sentence or statement that it has the minimum relative capacity of stomach in relation to body weight. So it requires more concentrates, as but its cecum is more developed. The ruminants do not have well developed cecum, and the equine or horses have well developed cecum, and there is also fermentation in cecum, but the ruminants are blessed with the compound stomach that gives them ample supply, ample space for the food to store and to digest to and that is and the equines are blessed with cecum. They have well developed cecum. Hormone villi indicates it will be related to small intestine. It is secreted from small intestine, as small intestine has villi, and they secrete the hormone villi. It is secreted by small intestine. And the horse is the simple stomach animal. So the horse has minimal relative capacity of stomach in relation to body weight, and horse has well developed cecum, and dog has the smallest size cecum. The less developed cecum is in dog, well developed cecum is in horse, and horse has a minimum relative capacity of stomach in relation to body weight. Hormone villicaline is secreted from small intestine, the process by which food particles are conveyed into mouth cavity is deglutition. Deglutition is the way of conveyance of food particles into the mouth cavity. Prehension is to hold the food or to hold the food or to hold the food, and the process by which the food particles are conveyed into the mouth. Food are holded as prehension, and they are entered into the mouth as deglutition. Parotid glands are serous glands. The type of parotid gland is serous, and the pH of gastric juice is more than P1. pH, they are acidic, but not that acidic that they can be less than 1, so they are acidic but not to that extent that their value may be less than 1. They are acidic but their value is more than 1. Conversion of it is pH of gastric juice is usually 2 to 3. Conversion of casein to para-casein is catalyzed by renin. Casein to para-casein. Renin converts casein to para-casein. Pepsinogen is activated to pepsin by the action of HCl. Pepsinogen is converted to pepsin by HCl in the stomach of animals and humans too. In glandular stomach, and its example is proventriculus in poultry, simple stomach in non-ruminant animals, and the abomasum in ruminants. So here the pepsinogen is converted to pepsin, the active substance, by the help of HCl, and the casein is converted to para-casein with the help of renin. Renin converts casein to para-casein. HCl converts pepsinogen to pepsin. pH of gastric juice is acidic 2 to 3, that is greater than 1, and serous glands, the parotid glands, are serous in nature, and deglutition is the conveying of food into the mouth cavity. Hormone will is secreted from small intestine, and the horse has a minimum relative capacity of stomach in relation to body weight, and it has horse has well developed cecum, and the less developed cecum is observed in dog. Blood platelets are increased in anemia. Blood platelets are decreased in thrombocytic purpura. Death due to sweet clover disease presents hemorrhages on postmortem, which if we see hemorrhages, then the sweet clover poisoning can also be the suspected one, or differential diagnostic should also be done with sweet clover poisoning. The formula of plasma volume into 100 divided by 100 minus 0.87 into hematocrit gives the blood volume, and the total number of blood groups found till now are 12 in cattle and 4 in humans. Humans have A, B, AB, and O. Which of the following helps him to keep iron in ferrous form? Globin. The globin of hemoglobin keeps iron in ferrous form, and this iron is responsible to carry oxygen for respiration. Which of the following is the for respiration? Which of the following is the organ of detoxification? Liver is the organ of detoxification. In parasitic infection, there is eosinophilia. Bone marrow and brain are deprived of lymphoid vessels, and lymphoid vessel flows towards heart. Which one of the following is responsible for secretion of hydrochloric acid HCl on stomach? Within stomach, parietal cells secrete HCl, and HCl is responsible for activation of pepsinogen to pepsin. HCl is secreted from the parietal cells. HCl is secreted from parietal cells, and this HCl is responsible for converting pepsinogen to pepsin for protein digestion, and renin converts casein to casein. Conversion of casein to casein, para-casein. Casein to para-casein is converted by renin, and the parietal cells of HCl are responsible for conversion of pepsinogen into pepsin, that is responsible for protein digestion. The center of vomission is located in hypothalamus. Vomission means vomit. The activation of vomission to initiation of vomit is by hypothalamus. Its center is in hypothalamus. Parietal cells secrete HCl. The conversion of trypsinogen to active trypsin is carried out by enterokinase. Enterokinase is responsible to convert the trypsinogen to trypsin, HCl from parietal cells is responsible to convert the pepsinogen to pepsin. Renin converts casein to paracasein. Renin converts casein to paracasein. HCl converts pepsinogen to pepsin, and enterokinase converts trypsinogen to trypsin. Hypothalamus center of formation is in hypothalamus, and parietal cells are responsible to secrete HCl. Match the following. Sertoli cells are in testes. Ketone bodies are beta hydroxybutyric acid. Ovulation fossa is in mare. Vitamin A is in carotene, vitamin A or carotene. Stercobilin is in feces. Feces has stercobilin. Vitamin A, carotene. Urobilin is in urine. Urine has urobilin, and feces has stercobilin. Both bilin means that they will be the product of breakdown of bile. Bilin indicates, and urobilin the product of bile in urine, and the stercobilin are in feces. The product of bile in urine is urobilin. Glomerular filtration pressure, or GFR, is the filtration coefficient. The functional efficiency of kidney or nephrons is called as the filtration coefficient or the glomerular filtration pressure. Non-granular leukocytes are lymphocytes. The granular lymphocytes are basophil, eosinophil, neutrophil. Neutrophils are the most predominant white blood cells of are the most prominent white blood cells in neutrophils, and they are approximately 50% of the total white blood cell count. And the non-granulocytes are monocytes and lymphocytes. Monocytes are in infection. Monocytes are modified into macrophages to undergo phagocytosis of the antigens, and lymphocytes are divided into T lymphocytes and B lymphocytes. B lymphocytes give rise to antibodies, and T cells give rise to cell mediated immunity as T helper cells, T cytotoxic cells, and interferons, etc. Cell mediated immunity is undergo by the T cells, and B cells has the humoral immunity or immunity by antibodies as they make antibodies, and both are the types are of lymphocytes. And non-granular leukocytes are lymphocytes. Granular leukocytes are eosinophil along with basophil and neutrophils. Neutrophils have 5 lobed nucleus. Maximum lobes are of neutrophils, and neurons have synapses. Sertoli cells are in testes. Ketone bodies are beta hydroxybutyric acid. Ovulation fossa is in mare. Vitamin A is carotene. Stercobilin is in feces. Urobilin is in urine. Glomerular filtration rate or filtration pressure is filtration coefficient. Non-granular leukocytes are lymphocytes. Granular leukocytes are eosinophils. The neurons have synapses, and enterokinase converts trypsinogen to trypsin. Hypothalamus is the center of vomtion. Parietal cells are responsible for secretion of HCl, and this HCl converts the pepsinogen to pepsin, and renin converts casein to paracasein. And next question is zymogens. Zymogen is pepsinogen. Pepsinogen is secreted from zymogen cells, and HCl is secreted from parietal cells. Parietal cells produces HCl. Parietal cells produce HCl, and zymogen cells produces pepsinogen. Honeycomb-like structure is in uh, honeycomb-like structure is in reticulum. Reticulum shape is has honeycomb-like structure. Zymogens secretes pepsinogen, and pouch of ruminant stomach is rumen. Rumen is the pouch of ruminant stomach, and the reticulum is the honeycomb structure like rennet of ruminant stomach is abomasum. Rennet of ruminant stomach is secreted from abomasum. Abomasum or rumen reticulum omasum. Abomasum is the true stomach, and it secretes rennet from ruminant stomach, and rumen is the pouch of ruminant stomach. Honeycomb like structure is reticulum. Juxta glomerular apparatus is Lacke's cells. Lacke's cells has juxtaglomerular apparatus. Rennet of ruminant stomach is secreted from abomasum. Pouch of ruminant stomach is rumen. Honeycomb-like structure is reticulum. Zymogen secretes pepsinogen, and HCl is secreted from parietal cells. Juxta glomerular apparatus in Lacke's cells. Lacke's cells has or Lacke's cells has juxtaglomerular apparatus. Origin of chela terminalis in cows is omasum. Omasum has the origin of chela terminalis in cow. Pregnant mare serum gonadotropin is a glycoprotein. Pregnant mare serum gonadotropin is a glycoprotein. Origin of chela terminalis in cow is omasum. Omasum has the origin of chela terminalis in cow, and the rennet of ruminant stomach is in abomasum. Chela terminalis and rennet. Origin of chela terminalis in cow is in omasum. The third part of the stomach and the rennet of ruminant stomach is in abomasum. The true stomach gives rise to rennet. Rennet is produced from the true stomach or the glandular stomach or the abomasum, whereas the origin of chela terminalis in cows is the third part of the ruminant stomach, that is omasum. It is also called as the book like. It is book like as it has many folds, so it is book shaped stomach. Pregnant mare serum gonadotropin is a glycoprotein. Prostaglandin is arachidonic acid derivative. Prostaglandin is the derivative of arachidonic acid. Pregnant mare serum gonadotropin is a glycoprotein. Prostaglandin is a derivative of arachidonic acid. Inhibin is the folliculostatin. Folliculostatin inhibin is a folliculostatin as it inhibits the luteinizing hormone, and luteinizing hormone is responsible for follicular as it inhibits the follicular stimulating hormone, and follicular stimulating hormone is responsible for the stimulation or the formation of or the maturation of the follicles, and inhibits that, so it is known as the folliculostatin. And gas analysis was developed by Haldane and Pistelli. Haldane and Pistelli developed gas analysis. And gas analysis was developed by Haldane and Pistelli. Inhibin is a folliculostatin, and folliculogenesis is by under follicle stimulating hormone, and ovulation is under LH surge, the luteinizing hormone surge. Prostaglandin is arachidonic acid derivative. Pregnant serum gonadotropin is a glycoprotein. Origin of chela terminalis in cow is the third part ruminant omasum, and the rennet of ruminant stomach is secreted from true stomach, that is the fourth part, or glandular stomach, that is the fourth part of the stomach, that is rumen reticulum abomasum. Abomasum is the uh, secretes rennet pouch of ruminant stomach is the rumen as the food first for the first time is forage for the first time is stored in rumen, and then it is regurgitated, and then it is processed and denatured into, or it is converted into its smallest or simplest molecule as carbohydrates, monosaccharides. Honeycomb like structure is reticulum. Reticulum has honeycomb like structure, and omesum has a book like structure, and it has many folds, partitions. Zymogen uh, zymogens are secreted from pepsinogen, and HCl is secreted from parietal cells. Match the before we had studied that Sertoli cells are secreted from testes. Ketone bodies are beta hydroxyl butyric acid. Ovulation foci is in mare. Vitamin A is from carotene, and stercobilin is from feces. Urobilin is from urine. Glomerular filtration rate or filtration pressure is filtration coefficient. Non-granuloleucocytes are lymphocytes. Granular leukocytes are eosinophil. Granular leukocytes are basophil, eosinophil, and neutrophil. Neurons have synapses. And match the following chemical digestion in birds. Chemical digestion in birds is in small intestine. Small intestine has the chemical digestion, or birds chemical digestion is in small intestine. Absorption of bile salts is in lower ileum. Lower ileum has the digestion of bile salts, whereas the upper ileum absorbs the amino acids. Upper ileum absorbs the amino acids, and lower part of the ileum absorbs bile salts. And next is same as that, absorption of endogenous amino acids is from the lower half of ileum. Absorption of endogenous amino acids is from the lower half of ileum. Amino acids are divided into exogenous amino acids and endogenous amino acids, and the endogenous amino acids are absorbed in lower half of ileum, and exogenous amino acids are absorbed in upper half of ileum. Ileum upper half and lower half. Firstly, the amino acid that is absorbed, or firstly, or the first part, or the first part of the ileum that the absorbs the amino acids are the exogenous amino acids. Exogenous amino acids are absorbed in the upper half, so we can say that exogenous amino acids are absorbed earlier and proceeded by the endogenous amino acids. Exogenous amino acids are absorbed first, and endogenous amino acids are absorbed later. Exogenous amino acids or external amino acids are absorbed first, and internal or body endogenous amino acids, they are absorbed later, but both are absorbed in the ileum. So if exogenous amino acid is absorbed first, so it should have to be absorbed from upper half of ileum, and if as the endogenous amino acids are absorbed later, so they should have to be absorbed on the lower half of the ileum. And chemical digestion of birds is in small intestine, and the absorption of bile salts is in lower ileum. Lower ileum has two functions, and upper ileum has till now one function, as upper ileum is responsible for absorption of exogenous amino acids. and the lower amino acids and the lower part of the ileum is responsible for the absorption uh, absorption of bile acids is in lower ileum. Point 1 is that lower ileum absorbs the bile acid and lower ileum absorbs the endogenous amino acids, bile salts and endogenous amino acids. Two things: endogenous amino acids and bile salts both are absorbed in lower half of ileum. Lower half of ileum is but the upper part of the ileum or the first half of ileum is responsible only for the absorption of exogenous amino acids, whereas the endogenous amino acids are absorbed along with bile salts in the lower half of the ileum. And inhibitor of leucine and phenylalanine is methionine. Poultry requires the amino acids. Major amino acids that are required in poultry are arginine, lysine, methionine, tryptophan, tyrosine, threonine. Arginine, lysine, methionine plus methionine, tryptophan, tyrosine. These are one of the major amino acids required by poultry, but poultry are majorly limiting at the arginine, lysine, methionine, tryptophan, tyrosine, as methionine is one of the important amino acids. So the point should have be considered that inhibitor methionine inhibits leucine and phenylalanine, or inhibitor of leucine and phenylalanine is methionine. And gastric pH of birds is it should be acidic, it is 0.5 to 2.5. That of mammals is slightly less than slightly more than that, it is 1 to 3, but mammals do not have usually the pH of 0.5. Birds has the pH of 0.5 to 2.5. And the bird stomach, true stomach, is known as proventriculus, and the glandular and the muscular stomach or the grinding stomach is the gizzard. Gizzard is the grinding stomach, and the glandular stomach that secretes HCl is the proventriculus, and its pH is 0.5 to 2.5. Intestinal pH of birds intestinal pH of birds is 6.6 to 8, slightly acidic to neutral to slightly basic. It is the pH of birds of intestine. And gastrine is pyloric stomach. Pyloric stomach secretes gastrine. The stomach can be divided into main part of the stomach is fundus, and fundus ends into the pyloric sphincter, and pyloric sphincter and that part of surrounding of the pyloric sphincter, the area of pyloric sphincter of the stomach is known as the pyloric stomach, and it secretes gastrine. Pyloric stomach secretes gastrine. Main body of stomach is known as fundus. Cholecytokinin and pancreozymin, they are secreted from upper part of small intestine. And remember that ileum is also part of small intestine, as the small intestine has three parts as duodenum, jejunum and ileum. Large intestine also has three parts as cecum, colon and rectum. Large intestine is well developed in equines for fermentation, whereas at that cholecytokinin and pancreozymin is secreted from the upper part of small intestine. It may be the duodenum or jejunum. It will not be the ileum, as ileum is the lower part of small intestine. So the upper part of small intestine is responsible for cholecytokinin and pancreozymin. Upper part of small intestine, duodenum and jejunum, whereas the last part of the small intestine as ileum, it is responsible according to these as three main secretions. As the lower part of the small intestine as the ileum, it can be divided into upper ileum and lower ileum, and the upper ileum is responsible for two main functions, that absorption of bile salts. The upper part of the small intestine, the upper part of the or the first part of ileum is responsible for the absorption of bile salts and endogenous amino acids, and the exogenous amino acids, they are secreted from upper half of ileum. Bile acids and endogenous amino acids, they are absorbed from lower ileum. We can say that at the lower part of the small intestine can be divided into upper part is the ileum, and ileum can be divided into upper half of the ileum and lower half of the ileum, and upper half of the ileum is responsible for only one main function, as the absorption of exogenous amino acids, whereas that of the lower part of the ileum is responsible for absorption of bile salts and absorption of endogenous amino acids. So in summary, we can say that the small intestine can be divided into two main, as the upper part of the small intestine and lower part of the small intestine. Upper part of the small intestine can be the duodenum and jejunum, and lower part of the small intestine can be the ileum. Upper part of small intestine is responsible for secretion of cholecytokinin and pancreozymin. Upper part of the small intestine and as duodenum and jejunum is responsible for the secretion of cholecytokinin and pancreozymin, whereas the lower part of the small intestine, that is ileum, can further be divided into upper part of ileum, upper ileum, first part of the ileum, or the lower part of the ileum, or that the ileum that is the last half. First half of ileum or upper part of ileum is responsible for absorption of exogenous amino acids, and the lower part of the ileum is responsible for the absorption of endogenous amino acids and the absorption of bile salts. So till now we have stated that zymogens are the secreted from pepsinogen honeycomb like structure is in reticulum. Differentiated lymphocytes producing immunologist. Plasma cells differentiates the lymphocytes, or the lymphocytes are actually the differentiation or modification of plasma cell. Eosinophil inclusions are known as curl of body are the eosinophilic inclusions, and plasma cell differentiates lymphocytes in actual are the differentiated plasma cell, or their precursor is plasma cell. Curl of body has the eosinophilic inclusion. Babesiosis in cattle has linked with Russell viper venom time. I do not understand that point, but according to that, babesiosis in cattle is linked with Russell viper venom time. Russell viper venom in actual is the venom of the Russell viper that has the ability to clot the blood, and it has been used in medical field also to clot the minor trauma etc where there is a bleeding. So it was also used to stop the bleeding in a minor cuts, abrasions etc, as the injury by surgical blade etc, or by the any other sharp object. Russell viper venom was used in past to stop the or to clot the blood. So it is linked with babesiosis in cattle. Anaplasma in cattle or anaplasmosis in cattle has extravascular hemolysis. Anaplasmosis or anaplasma marginal as an example causes extravascular hemolysis, and babesiosis in cattle is linked with the Russell viper venom time and eosinophilic inclusion. Eosinophilic inclusion is in curl-off body, or curl-off body has the eosinophilic inclusions. Anaplasmosis in cattle has extravascular hemolysis, red blood cells with no central pallor. It means center is not pale and smaller than normal. The red blood cell that is smaller than normal and it is not pale or faded reddish from the center. It is the spherocytes. Spherocytes or spherocytes are the red blood cells that are not fade from center or pale from center or paler center and are smaller than the normal size. 16 micrometer is the size of the red blood cells of sheep, goats. Red blood cell has 16 micrometer, and if the goat having the less than 16 micrometer size is red blood cell is less than 16 micrometer, and it is not paler from the center or not faded color, not a reddish color, no reddish color, and it has the reddish color. It is not paler or it is not faded. The red blood cell is less than 16. In case of goats, the red blood cell is less than 16 micrometer, and it is not paler or not faded from the center. Then it will be the spherocyte. Spherocyte is the red blood cell with no central pillar and smaller than normal. Anaplasmosis in cattle has the extravascular hemolysis. Result viper venom time is linked with BVSc in cattle. Eosinophilic inclusion are in spherocytes, or spherocytes are the eosinophilic inclusion, have the eosinophilic inclusions. Differentiated lymphocytes are differentiated from plasma cells, and fragment of red blood cells are schistocytes. Schistocytes are the fragmented red blood cells. We stated earlier that schistocytes are the fragment of red blood cells, and spherocytes are the schistocytes are the fragmented red blood cells, and spherocytes are the smaller, no central pillar red blood cells. The difference between spherocytes and schistocytes. Schistocytes are the fragmented, and the spherocytes are smaller and no pillar central. And test using recipient's plasma. In case of major cross or minor cross, we have to only remember that we have to remember the major cross. And in major cross, if we are the donor and we are donating our blood to any patient, then if our cells are being considered or tested, then it will be the major cross. Major cross will be the cross if the donor's cells are being tested. So from 112 and 13, test using recipient's plasma and donor cell. If I am the donor, my cells are being considered, then it will be the major test cross. And the test using donor plasma cells and recipient cells, if I am the donor, my cells are not being considered, so it will be the minor test cross. Immunoglobulin filled vacuole in cytoplasm. Vacuole in cytoplasm of plasma cell. If the plasma cell has a vacuole and this vacuole has the immunoglobulin, then it will be the Russell body. So the difference in spherocyte, schistocyte, and Russell body is the Russell body is not the red blood cell linked with red blood cell. Russell body is linked with plasma cell. If the plasma cell has a vacuole and this vacuole has immunoglobulins are of 5 types: G, A, M, D, E. So these are the 5 types of immunoglobulin. If any of them immunoglobulin is within the vacuole of the plasma cell, then it will be the Russell body. And the difference between spherocyte and schistocyte is that schistocyte is simple, the fragmented red blood cell, and spherocytes are the red blood cells that are smaller than the normal. For example, if the normal size of the red blood cell of goat is 16 micrometer, if the red blood cell is smaller than 16 micrometer, 12 micrometer or 10 micrometer, and it is not paler from the center, then it will be the spherocyte. And anaplasma in cattle is causes extravascular hemolysis. BVSc in cattle is linked with Russell viper venom time. Russell viper venom is famous for clotting the blood. Eosinophilic inclusions are the curl-off body are the eosinophilic inclusion. It should also not be confused with spherocyte, schistocyte, or Russell body. Eosinophilic inclusion, it is related. Eosin means Eosin means red loving, it will be the blue color cells. Basophil means basophilic cells are seem to be reddish. Eosinophilic cells seems to be the eosinophil seems to be bluish, and basophilic seems to be the reddish color. Eosinophil loves eosin, basophilic loves basophil. If basic color blue is the basic color, blue loving it will be the red. Eosinophil, they are the bluish color. So yeah, bluish color inclusions are in curl of body. Curl of body is the eosinophilic inclusion. Differentiated lymphocytes producing or simple plasma cell is differentiated into lymphocytes, or the lymphocytes are the precursor of lymphocytes is the plasma cell. Next, hemoglobin contains 4 atoms of iron and 8 atoms of sulfur. The amount of sulfur is double to that of the iron in hemoglobin. So iron is important as oxygen binds with the iron. So one molecule of hemoglobin has 4 globin molecules. Each globin has an iron molecule, and this iron is responsible to bind the oxygen. So a hemoglobin can bind 4 oxygen molecules, and but despite of that fact, the amount of sulfur is double than that of the amount of iron, and it is 8 atoms of sulfur. And onion is a rich source of sulfur. So onion may have the importance with that the chemistry of red blood cell, that the red blood cell has the 4 atoms of sulfur. The molecular weight of hemoglobin is 68,000. 68,000 is the molecular weight of hemoglobin. We can remember it with that fact that the 8 molecules are of sulfur. So 8 number can be remembered from that fact. And the pig has rapid growth and poor iron reserve, and it has increased iron requirement for erythropoiesis. Pigs require the fortification of iron or the external supplementation of iron, whereas the ruminants and other animals, they usually do not require ruminants do not usually require the extra supplementation of iron, whereas the pig requires the extra supplementation of iron as it has the rapid growth is also in other animals and ruminants too, but it has a poor iron reserve. Pigs have the poor iron reserve, so they require the fortification or the iron and to be provided from the diet. Plumbism is the poisoning of lead. The lead is denoted as Pb, and the poisoning of Pb is plumbism. Anaplasma marginalis, an intracellular parasite, is referred to as marginal bodies. As the name indicates, anaplasma marginal means it will be on the margin, so it is also referred to as the marginal bodies in the red blood cells. Aplastic anemia is associated with Bracken-Fern poisoning. Bracken-Fern poisoning leads to aplastic anemia that may lead to death. And there are also the hemorrhages in Bracken-Fern poisoning. On postmortem examination, if we see the hemorrhages, then we can suspect it for Bracken-Fern poisoning, and we can differential diagnose. Another diagnostic pointer is that there will be the aplastic anemia. Basophil and mast cells are the source of indigenous heparin. Basophil is a type of white blood cell that is granulocytic white blood cell. Other type of granulocytic white blood cells are basophil, eosinophil, and neutrophil. So basophil along with mast cells are the source of indigenous heparin, and heparin is the blood clotting clots the blood, is responsible for clotting the blood. So basophil and mast cells are the source of indigenous heparin. Aplastic anemia is associated with Bracken-Fern poisoning. Bracken-Fern poisoning also causes the hemorrhages, and in deer, the normal shape of red blood cells in deer is sickle shaped, whereas the sickle shaped in humans may lead to the sickle cell anemia, a disease. So sickle cell in red blood cells in deer, it is a normal phenomena. Their normal cells are sickle shaped, and deer sickle cell erythrocytes is naturally occurring, Morphological variation, it is the normal cells of deer. Red blood cells of deer are of a sickle shape. The plasma protein fibrinogen is used to detect inflammation in large animals, or simply fibrinogen is the inflammation detector. Inflammation can be detected in large animals by detecting simply the fibrinogen. More the fibrinogen, more is the inflammation; less the fibrinogen, less is the inflammation. So inflammatory response detection can be through the fibrinogen concentration. Blood proteins are produced by hepatocyte, leukocytes, and vascular endothelium. Vascular endothelium is the lining of the vessels. So blood proteins are produced by the blood carrying system or the vascular endothelium. Hepatocyte cells of the liver, and leukocytes, the white blood cell leukocytes, as the that the lymphocytes can be differentiated into T cells and B cells. T cells secrete cytokines or inflammatory mediators, and these inflammatory mediators are as interferon, gamma, temporal necrosis factor, interleukins. There are also the blood proteins, and they are secreted from leukocytes, and special leukocyte that is involved with in inner cell regulation against the pathogens is by the cell mediated immunity, by the proteins as interleukin, interferons, tumor necrosis factors, etc. So hepatocytes are also involved in blood protein synthesis. Vascular endothelium is also involved in blood protein synthesis. Plasma protein fibrinogen is the inflammation detector, can be used to detect inflammation. Deer has sickle-shaped erythrocytes. Basophil and mast cells are the source of indigenous heparin. Heparin can be obtained from basophil and mast cells. Aplastic anemia is associated with bacon fern poisoning. Smooth endoplasmic reticulum is the major source of intracellular storage set for the element calcium. Smooth endoplasmic reticulum is the major the calcium is stored intracellularly in the smooth endoplasmic reticulum. Calcium is stored within the cell in the endoplasmic reticulum, and especially the smooth endoplasmic reticulum. The movement of lipid soluble substrate through the cell's membrane is by the process of diffusion, or the movement of cells without movement of substrate or anything against the concentration gradient is by active transport, and the movement of anything from the region of higher concentration to the region of lower concentration is the diffusion. So the lipid soluble substance through cells, at the cells itself they are the lipid bilayer. So lipid can pass through the lipid, so the lipid soluble substrate through the lipid membrane by process of diffusion, as though no energy is required. Spleen is called the graveyard of red blood cells. Spleen is the graveyard of red blood cells. Gallbladder is absent in horse. Horse do not have the gallbladder, and gallbladder is the storage site for bile. Bile is synthesized in liver. Bile is synthesized in liver and is stored in gallbladder in the animals that have the gallbladder. Horse do not have gallbladder, so horse cannot store its bile. Whereas humans can influence, they can store their bile that is secreted from liver. The specific gravity of bile is 1.010. 1.0 or approximately 1 is the specific gravity of bile that is secreted from liver and stored in gallbladder, and gallbladder is absent in horse. Bile is bitter in taste. Taste of bile is bitter in carnivore. Bile is golden yellow color. Carnivores bile is golden yellow color, whereas the humans and normally bile is greenish in color. But the golden yellow color or yellowish color, bright yellow color, this color that is same as that of the sun color is the color of bile in carnivores, as lion, tiger, leopard, etc. Cholerectic increases secretion of bile. Cholerectics can be used to increase the secretion of bile during rumination, and bile is I think associated with the digestion of lipids, but not sure. And the bile is secreted from cholerectics during rumination. The number of ruminoreticular cycles per minute is one cycle per minute is the ruminoreticular cycle. Ruminoreticular cycle is 1 per minute. Cholerectic increases bile secretion. Bile color is golden yellow bile is bitter in taste, and the naturally placenta is expelled within 4 to 8 hours of parturition. In cattle, loss of 7 to 10% of body weight results in death. Chloride shift is also known as hamburger phenomena. Hamburger phenomena is associated with red blood cells in which the chloride enter or exit the red blood cells by this hamburger phenomena. Loss of 7 to 10% of body weight can result to the death, and the normal placenta is expelled within 4 to 10 hours of the cattle. During rumination, the number of ruminoreticular cycles per minute is 1. Choreotics are the drugs that increase the secretion of bile in carnivores. Bile is golden yellow colored. Bile is bitter in taste. The specific gravity of bile is 1. Bile is synthesized in liver. Gallbladder is absent in horse. Spleen is called the graveyard of red blood cells, and diffusion is the movement of molecule from the region of higher concentration to lower concentration without use of energy. Smooth endoplasmic reticulum is the major storage of calcium. Calcium is stored in the cell. In the smooth endoplasmic reticulum, the blood proteins are hepatocytes, leukocytes, and vascular endothelium. The plasma protein fibrinogen is used to detect inflammation. Inflammation can be detected by fibrinogen. Deer red blood cells are sickle-shaped. Basophil and mast cells are source of indigenous heparin. Heparin is the blood clotting protein, aplastic blood anticoagulant. It is anticoagulant. Basophil and mast cells are secreted from that. Heparin is the heparin is the blood anti-clotting protein. Aplastic anemia is associated with Bracken-Fern poisoning. Bracken-Fern poisoning causes hemorrhages. So if there is any animal that is suspected and for any poisoning and have hemorrhages and aplastic anemia, then it will be have the Bracken-Fern poisoning. This should also have to be differentiated diagnosis. Anaplasma marginal is also referred to as having the marginal bodies. Red blood cells having marginal bodies can be suspected for anaplasma marginal. Plumpism is the poisoning of PB or lead. Pig has a rapid growth rate and is deficient of iron, or it cannot store iron, so it requires the fortification or the external diet having the iron. Weight loss, the molecular weight of hemoglobin is 68,000, and the hemoglobin has 4 iron and 8 atoms of sulfur, and the hemoglobin can bind 4 oxygen with 4 iron of the 4 globin molecules. Differentiated lymphocytes are from the plasma cell. The eosinophilic inclusion is the curl of body. Curl of body is the eosinophilic inclusion. BVSc in cattle is the Russell Viber venom time. Anaplasmosis in cattle leads to extravascular hemolysis. Red blood cell with no central pillar and smaller than normal is the spherocyte. Spherocyte is the red blood cell with no central pillar and smaller than normal. Fragment of red blood cell is the schistocytes. Immunoglobulin filled vacuole in the cytoplasm or plasma cell having the vacuole, and this vacuole have immunoglobulin. Then this cell will be called as this plasma cell will be called as a Russell body. Russell body is not the anomaly of red blood cell. Remember that mostly anomalies are of the red blood cells, very less are as the Russell body is the anomaly of plasma cell. And the eosinophilic inclusion, as it is the white blood cell linked, it is called as curl of body. Immunoglobulin and the test using plasma, major test cross and minor test cross. Major test cross is the test cross in which if I am the, for example, I am the donor, I want to donate blood to anyone, and if my cells are being tested, then it will be the major test cross.